Okay, so in today's video we are going to have a look at the topic of meiosis and variation. So there you go. Nice picture courtesy to my daughter, thank you very much. So specifically we are looking at question 5 of the assignment to cell division, and within this question you have several parts. The first part is asking you to compare mitosis and meiosis. So I'm going to leave this part of the question for you to do independently. Now, you can easily find these sort of tables in books or websites where mitosis and meiosis get compared. One suggestion I would say is try to expand on this, on each of the points a little bit. So for example, if you're looking at the number of chromosomes in the resulting cells, instead of just, I don't know, saying 46 chromosomes as opposed to 23, just sort of make it into a sentence, just make a full statement. So what is that number, what does it mean, and what it refers to? Okay, so that's one bit of advice I'd give for higher grades. So, but today what I'm specifically interested in is which mechanisms within meiosis produce genetic variation. So which mechanisms in meiosis produce genetic variation. Okay, so that's the question I'm posing. So just a reminder, and I hope a lot of you got to the topic of meiosis already, but for those of you who maybe haven't quite got that far, or you want a bit of a sort of recap, in meiosis what you are doing is you're producing sex cells. So if you remember from my previous video, sex cells mean egg in a female and sperm in a male. Okay. So you only ever produce sex cells, so no other cells are being produced through the process of meiosis. So in terms of sex cells, so let's think how sex cells are different to any other cell in your body. So in which way are they different? In which way are they different? So there are two main sort of points of difference between sex cells and any other cell in your body, say your brain cell or your skin cell or your liver cell. So the first point of difference is that they contain half the number of chromosomes. So they contain 23 chromosomes. And I'm talking specifically about humans here, right? Because other species will have their own different number of chromosomes. But when it comes to humans, you're going to have 23 chromosomes either in an egg or in a sperm cell. So 23 chromosomes. And this number, I hope you still remember from the first video, that number is known as haploid number. So this is haploid number. Now, the other way in which sex cells are a little bit different is that each single sex cell is completely and utterly one-off. So it's totally genetically unique. So it's genetically unique. So never seen before, never to be repeated again. So every single egg in the ovaries in the female and every single sperm in the testes in a male is totally different to the next one. And that's what sex cells are, and that's what needs to be achieved, therefore, during meiosis. You have to achieve these two objectives. You have to produce cells which are half the chromosome number, have haploid number of chromosomes, and the ones which are also all completely different to the next sex cell. So what I'm going to do is I'm going to look at which processes specifically produce that genetic variation. What is it which happens during meiosis which makes those sex cells completely unique and completely different to each other? So there are two main processes in meiosis which allow for that genetic variation to occur. So the first process which happens is called crossing over. And this process only happens in meiosis, and it does not occur in mitosis, right? Because there you're trying to produce identical cells, right? So therefore, crossing over is unique to meiosis. So what happens during crossing over? So I've already have a little diagram for you to show. So this is a diagram summarizing crossing over. So let's say you've got a pair of chromosomes, and they have to be homologous chromosomes. So this is a pair of homologous chromosomes. Right? If you're not sure what homologous chromosomes mean, you need to go back and rewatch the first video, have a look it up online, say. So here I've got two chromosomes. So one chromosome from the pair came from a mother of this person who is producing sex cells, and the second chromosome, let's say that dark green chromosome, came from the father of this person. So what happens during crossing over? Those chromosomes get really close together, so they need to be physically really, really sort of almost like touching, you know, side by side like this, okay? And they swap little fragments between each other, right? Now the fragments which have to be swapped, they need to be located on non-sister chromatids, right? So let's try to recall what that means. So it means that those sides, the shoulders of chromosomes, they need to belong to two different chromosomes. That's what non-sister chromatids mean. So let me just write it down. So they're non-sister chromatids. So the bits which are exchanged, they need to exist on non-sister chromatids, because sister chromatids would mean just those two sides of the same chromosome. So if I swap pink with another bit of pink, well, that wouldn't have achieved anything. So I need to be swapping non-sister chromatids. And on my diagram, I'm showing that two sections are getting swapped. So I've got one section here getting swapped and another section on this side of the chromosomes which are getting swapped. And they need to be equivalent kind of length and equivalent location, because if smaller part of a chromosome, let's say like a tiny bit of a green one, would be swapped with quite a big chunk of a pink one, that would cause something called mutation. So that would be a mistake. So one of the chromosomes would end up with too much information and another would end up with too little. So that's not good news. So they need to be exchanged in kind of equivalent parts, equivalent sections. So the point of doing that is that you started off with one pair of chromosomes where one whole chromosome was completely information from the mother of this person and another one was completely from the father. But by the end of crossing over, you got a situation where you've got two chromosomes and they just have a bit of everything, a bit of information from the mother and a little bit of information from the father. And the most important thing about this process is that this process is completely random, right? So it's totally random. So you cannot predict which particular part of which chromosome is going to get swapped. And because you can't predict it, every time two chromosomes get together during crossing over, the outcome is completely unpredictable. You just don't know what's going to happen. So it might be the case that just one fragment is going to get exchanged It might be the case that 2 fragments are going to get exchanged the way I'm showing on my diagram here, or it might be that hundreds of really tiny fragments are going to get exchanged. So you really don't know what you're going to get. And that, as you can imagine, is going to contribute to variation, to kind of that unpredictability about what the outcome is going to be. So that's my first kind of mechanism of producing genetically unique cells. Now, another important information you need to know is that process, that whole thing, it happens in the stage of meiosis, which is called prophase 1. Okay? And for those of you who are not aware of it, the 1 I'm using is the Roman numeral 1 because that's how it's traditionally written for. So meiosis 1 is going to be meiosis Roman numeral 1, and the stage within meiosis 1, which is called prophase 1, this is when crossing over occurs. Okay, so that's the first process which is generating genetic variation. So then you've got the second process. So let me move this piece of card out of the way. And so I'm going to write down my second process. Now, what I also would advise you, actually, just occurred to me, what I'd like you to do is potentially pause this video. And let me just come back to this picture. And just looking at this picture for crossing over, write down how you understand this process. So basically, describe to me what you can see on this picture. So that's what I would recommend. So pause now, have a look at this diagram. You've got all your labels, and write a short paragraph, maybe 2, 3, 4 sentences, just explaining what's going on this picture. Okay? Okay, so welcome back. I hope you managed to write down your little description of crossing over. So now it's time to move on to the second process, which contributes to genetic variation. So the second process is called independent assortment of homologous chromosomes. I know it's a bit of a mouthful, isn't it? But I'm going to write it down for you. So it's independent assortment of homologous chromosomes. Right, there you go. So independent assortment of homologous chromosomes. So I've got then my next diagram. This is going to look sort of pretty complicated to start with, but bear with me and I'll try my best to explain it. So to make it kind of more easier to follow, I'm going to cover part of this picture to start with. So as you can see on my picture straight away, I've got a bit of a label going on, and it says metaphase equator or plate. So that should tell you that this process is occurring in the stage called metaphase. And specifically, it's occurring in metaphase 1. Okay, so again, first division of meiosis 1, name of the stage is metaphase 1. Again, I'm using Roman numeral here. So what's going on in metaphase 1 is the pairs of chromosomes which exchanged fragments earlier on. So remember those ones, yeah? So that's what I'm drawing. So they've already exchanged fragments, that pair of chromosomes. And those pairs of chromosomes, they line up on that midpoint, so-called metaphase equator or metaphase plate. So your pairs of chromosomes are lining up along this equator. And on my first picture, you can see here, I've got mostly pink chromosome on this side and mostly red chromosome on this side. And then on the other side, I've got mostly dark green and blue chromosomes. So let's now say that the pink and the red chromosomes originated from the mother of this person, and the green and blue came originally from the father. I know, fair enough, it's got a little bit of swapping, which happened in crossing over. But I would say predominantly this side of the cell sort of is mostly information coming from the mother, and that side of the cell is mostly information coming from the father. Now, what could also happen is those chromosomes could actually swap places. They can't swap places like that because they were crossed over, so they're still kind of together at this point. But what could happen is that they can align along that midline in a different orientation. So let me show you the second picture. So my second picture, the first pair of chromosomes are exactly the same. So I still have pink one on this side, green one on this one. But the second pair of chromosomes, they swapped places. So the one which is mostly blue is now on the other side of the equator, as you can see. So what that would mean is that as meiosis 1 carries on happening, in the next stage, those two chromosomes are going to get drawn to this side of the cell. The other two chromosomes are going to get drawn to the opposite side of the cells, and those cells are going to split in half. So the end result of this whole process is going to be, in this scenario, right, you've got mostly pink and red chromosomes together, and here mostly green and mostly blue together. And in my second kind of option, it's the pink and the blue, which would be grouped together, and the green and the red would be grouped together. So, well, you might ask, well, so what's the big deal? You know, why does it matter? Well, let's sort of try to link this to real life. Let's now say that this particular sex cell ended up being the result of, being used to produce a baby, right? I mean, we still need to kind of undergo the second stage of cell division, but let's say whatever, came out as a result of that particular cell was beginning of somebody's life, right? That was our baby. And so my family came together, they all gathered around this baby, and they're having a look at this baby, and they're saying to each other, look at this wonderful baby, let's call her, I don't know, Katie. So let's look at Katie. Oh, wow, she looks just like her grandma. And guess why they're saying it? Because her chromosomes were grouped where you have mostly chromosomes coming from the mother of the person who was producing the sex cell, so grandmother of this little baby. And if the other sex cell, this one, ended up producing a different child, right, ended up being used for, conception, and that was resulted in a successful pregnancy, you would have completely different outcomes. So, your family would, again, gather together, have a look at this wonderful baby, and say, oh, look, Katie looks just like her granddad. And that's because she mostly has information from that side of the family, mostly from the father of a person who was producing those sex cells. So that would be grandfather of our little child. Now, in this scenario, your outcomes would be completely different, as you can see, right? So you have a lot more of a kind of mix going on between the mother's side and the father's side. So that sort of flipping of chromosomes can have pretty dramatic effect on the outcome and on the combination of genes that the resulting child is going to inherit. And so this process, as I mentioned before, is called independent assortment of homologous chromosomes. So let's do just a little bit of maths, a little bit of calculation here. So I'm going to move this card just a tiny bit out of the way. So mathematically, you can actually work out how many possible combinations you are going to, end up with as a result of independent assortment. Now, on my picture, I only have 2 pairs of homologous chromosomes showing. But in real life, I hope you're aware of it, you actually have 46 chromosomes or 23 pairs. So the same sort of principle is going to apply to 23 pairs of chromosomes when you're producing an egg or a sperm. So let's, as I said, put a little bit of calculation forward. So I've got 2 chromosomes in each pair. That's fairly obvious. And if I just pick one single pair, then that becomes 2 in the power of 1, which is just 2. And if I have 2 pairs of chromosomes, so that's what I had on my picture, right? So it's right there. So 2 chromosomes in a pair, and I've got 2 pairs of chromosomes, so that means 2 multiplied by 2. That's how 2 in the power of 2 works. And that just means 4. So I have 4 possible outcomes, right? 4 possible kind of orientations of 2 pairs of chromosomes. And here I've got 2 outcomes when I had just a single pair. And if I had 3 pairs of chromosomes, right, so still 2 chromosomes in a pair, 3 pairs of chromosomes, that would mean it's 2 multiplied by 2 multiplied by another 2, which would give me 8 possibilities, 8 outcomes. And I could carry on that calculation and finally arrive to the fact that actually I don't have just 1 pair or 2 pairs or 3 pairs of chromosomes. In real life, I have 23 pairs of chromosomes. And the lesson actually encouraged people to do a bit of calculation. And again, if you haven't done it, I would suggest you pause the video at this point, grab a calculator or just your phone and have a go at calculating for me, what is this number? So all you have to do is just multiply, keep multiplying 2. So it's 2 by 2 by 2 by 2 by 2 by 2, and you do the same thing 23 times, right? So don't do 23 multiplied by 2. That's not going to be right. So it's 2 multiplied by itself 23 times. Okay, so pause now, have a go at calculating this number, and let's see if your number is going to be the same as my number. Okay, so I hope you had a go at it, and I actually, to be honest with you, I haven't got this number in front of me, but from memory, what you should have is around about 8.4 million, or just under 8.4 million possibilities or outcomes, right? So around about, I think it's 8.3 something something something. So it's just around. By if I round it up, I should come up with 8.4 million. So by just flipping those chromosomes over, I can create over 8 million possible combinations. So that would be like having a really big combination lock with 23 little wheels, and every time you just twist one wheel a little bit, that would give you a different possibility. So that's another mechanism during meiosis which causes genetic variation, which results in cells which are genetically unique and never seen again. So when you combine the 2 processes together, crossing over, which is a random process, and independent assortment, which is another random process, really you get pretty much limitless number of combinations. So these 2 processes, right, crossing over and independent assortment, both of them happen during meiosis. Okay. Now there is third process which happens afterwards. So this happens after meiosis, but I think it would be kind of worth, mentioning it as well. So that's number 3, and it's called random nature of fertilization. It's called random nature of fertilization. So in this kind of process, what happens is you just have to kind of look at which egg is going to get released or which sperm is going to get released, and the reality is that it's again just completely and utterly random process. It's totally random. So let's just put it into kind of some numbers, right? So let's think about specific numbers. So when it comes to female eggs, so female females typically born with roundabout, and again, depending on which source of information you're going to look at, it might be slightly different. But I'm going to just use one of the sources I usually use, and that number is round about 400,000 eggs. So this is at birth. Right, so this number is going to start dropping as the female is getting older. But we're talking, let's sort of take that number as a starting point. So what random nature of fertilization says is that you cannot predict which particular egg out of that selection of 400,000 is going to get released, and you know which particular egg is going to be successful in potentially producing a baby. Now, for a male, this number, so male sperm, obviously, this number is even more staggering. So we're talking about, so this is not just at birth, but per ejaculation, no, I did it right first time, ejaculation, there you go. So per ejaculation, we are talking around about 200 to 500 million sperm cells. Right? So huge number, right? So huge number. So we're not even talking about sort of lifetime supply, but we're talking about just per one go, right? And again, you know, you cannot predict out of that staggering number of sperm cells which one is going to be successful. This is again just completely and utterly random. So that's another way of making sure that because you can't predict which egg and which sperm is going to be released, and you know which combination of egg and sperm is going to be successful in producing embryo, that just gives you another layer of randomization, of making sure that every outcome, every child of a specific couple is different to the next one, and that's what variation is. You know, it's just basically what makes us unique. What makes each of us is, you know, different person to the next person. On this planet Earth. There is obviously one exception, which is identical twins, but absolute majority of people obviously totally genetically unique, and you know their combination of genes cannot be repeated exactly. Okay, so that's the end of this video. In my next video, I'm going to look at why variation is important. So why did nature, bother, if you like, in producing all this staggering variation? Why can't we just all be clones and be happy and just live like all of us could be just sea of identical twins? So why do we need variation? So I'm going to discuss it in my next lecture, but for now, thank you very much and goodbye.